Hi, Anne.
Hi, how are you?
Good to see you. Good. Looking forward.
Thank you.
So Malte and Steph will come in soon as well.
Okay, sounds good.
And let me check the number of registrations. Yesterday it was 22. Um, given the no-show rate, the usual no-show rate, I wouldn't get my hopes too high.
Okay.
But now it's 24, including you. So, and I resent the invitation yesterday, so
Okay.
people should have gotten a reminder. But yeah, however many or few people come in, we hope to create a meaningful conversation, and our main focus will be to direct the conversation towards generating leads towards projects for you. But yeah, we will see what happens.
All right, sounds good.
No promises. Yeah. I'm gonna let the others know that you're here. Are you excited about the 20th? It's gonna be.
I can't come soon enough.
Yeah.
Yeah, I feel like I still always kind of feel like I don't really feel confident that it's gonna happen until it happens. Right? Finally, if Trump is gone and Biden is in, then I'll feel a bit,
And that it's went by positively and safely. Yeah. I mean,
indeed.
Now that the guy's deplatformed, it's probably a little bit harder to communicate to these strange, crazy people.
Yeah, indeed. I hope so.
But you never know.
Yeah, it's funny how it doesn't occur to him to just go down to his, in his own house. There's a room with reporters. He can go there whenever he wants, But that's not as attractive to him as, deranged tweets that he makes.
Well, he might do that. Then the question is, do they report what he says?
Yeah, I was surprised to see that Angela Merkel didn't agree with the idea of kicking Trump off of Twitter. It's funny because I always respect Germany so much for removing the voice of neo-Nazis, right? Like saying you can't use the symbols, you can't discuss it, right?
I had the same notion. Totally, I had the same notion.
I would have thought that most Germans would approve of kicking Trump off Twitter, but
yeah, I agree.
Hi.
Hello.
Hi, how are you?
Good. How are you doing?
Good.
Good. Are you speaking about the current situation? Is he going to be impeached? Yes, no? Oh dear. That's going to be exciting. Thomas, do you want me to send Malte the link again? Because he was just asking whether he's in the wrong meeting.
Yeah.
He sent me this link.
I know. I'm actually going to resend it to him.
Yeah, I'm copying it as well.
It's done.
We got it. So now he's. Aravind.
Hi.
Good to see you. Long time.
Hi. Yeah, just in one client location, so I thought that I could quickly just say hello to you guys and Hi, good to meet you, Anna and
Hi, hello.
Stephanie, right?
Exactly.
Yeah.
Hi,
Okay.
Arvind.
Yeah.
Where are you in Berlin at the moment?
Yeah, in Berlin. Yeah. So this is one factory and it's completely locked down. So no factory. I mean, there's some people for such a big size. It's some people are there and seriously affected their operations. Yeah. And so how many participants are we looking
Well,
for?
24 people registered, and given the usual no-show rates, I would expect about half of those. And now our
Malte.
boss and moderator is on board as well. Hi, Malte.
Hello, everyone. So I was actually in the wrong in the meeting room. I was in your personal meeting room, Thomas.
Oh, no.
And nobody was
I
there.
hope it was nice. I hope maybe you met somebody.
Yeah, it was quite cozy. So,
okay. So I would suggest we wait a couple of more minutes. We're still too early for everybody to come in. Edgar, where are you located?
Hello.
Hello.
I'm located in Latvia, in Riga, backgrounds, yeah. So nice to meet you all.
Nice to meet you too.
Yeah. I'm doing some other job as well, so that's why I'm switching off the camera.
Okay.
I think it's fine.
Yeah, don't worry.
Thanks. Thank you.
Malte, maybe you want to share some music or some thoughts. It's always the situation at the beginning.
Waiting for everyone else because we're too early. Aravid, I want to thank you that you shared everything yesterday. Get some quick response by some people on LinkedIn, and I shared it till, I don't know, 1 in the evening with some people.
Okay, no worries. I mean, it was a little bit, should have been done a little bit earlier, but yeah, things got missed and good at least in the last moment.
Welcome, Arman.
Hello, Herman.
Hello, good afternoon to all of you. Good evening, depending on which part of the world you are in.
Good morning.
Good morning.
Hi, Herman.
I think Anne, what's your time, Anne?
9 in the morning.
Okay.
Well, good morning to you then.
So Herman, we're still waiting for a couple of people.
All right. I'll be with you in a moment.
Malte, I will mute myself now, so and head over to you.
Actually, I was looking for some music, but I don't know how to play it here. So, hello, TJ. So, hello, Matthew.
Hi, hello. Good afternoon, everyone. Thanks for inviting me.
Hello, where are you? Thanks for having you. Where are you coming from?
I'm from Belgium.
Oh, nice. And also worked in Belgium in the Netherlands.
I was, yeah, the Netherlands.
So.
Yeah, I was in Delft for almost 8 years at TU Delft.
Oh, I lived in Delft for 2 years.
Really?
Yeah.
Okay. Work Patria. Okay, shall we wait 1 minute more? And I'm trying to figure out how to use Zoom. So, okay. Okay, let's start. Okay, first of all, welcome everyone. Today we are hosting our first event of NextNess, which is all about regenerative materials. And it all started in the beginning last year when we thought about, okay, what can we actually do for sustainable development, and how can we actually create a world where we don't extract materials, but we rather create them. And me, Thomas, and Stephanie, we thought about, okay, we have like big factors in disrupting sectors like in energy, transportation, and also materials. And me personally, I believe that materials will be one of the largest disrupt sector in a couple of years. And usually we don't really think about materials in a sustainable way, rather in a very like unsustainable way. And so we thought about, can we actually create living materials which have characteristic of biological systems like self-replication, self-regulation, self-healing, and self-sustainable. So we reached out to a couple of researchers last year and start talking about the subject. And one of those researchers was Anne, and I was very happy to start talking with her here about living materials and what can we actually do with bacteria. And then we thought, okay, maybe it's a very good idea to start a conversation about that and bring more people into that, where we can start and talk about, like, is it possible to scale materials, and where we have actually a room or space where we can talk about materials, and also like from different perspectives with researchers and also investors and startups and those kind of things. So the agenda for today is so I introduced NextNess a little bit. I will take in Anne in a moment, and a little housekeeping beside that. We have a chat window where you can ask questions, and we will collect and curate them and ask them later in the Q&A. And I would turn off all the cameras for a better recording quality. And I will introduce Anne in a moment, and then we will start with presentation about living materials, and afterwards we will start talking about Q&A and what's actually possible within the next 3 years. So as I said, I found Anne last year, and she's a professor in biology at the University of Rochester. She received her PhD in biology science from Stanford, right? And no further introduction. Here's Anne, and let's see how I can unmute you. Oh,
I don't have to do it. Unmuted.
Hey.
So thank you to Malta, Thomas, and Stephanie for this invitation. It's great to be the, kicking off this NextNess series. Let me work on sharing my slides. All right, is that looking good? And a thumbs up from Malta if it looks all right. All right, perfect. Right, so as Malta talked about, I'll be talking today about something my lab is working on to see if we can produce materials using biological and synthetic biology inspired techniques in a way to make materials that are really environmentally friendly in the way we make advanced materials that can have, interesting shapes and interesting functions. So, when I started my lab 10 years ago now, I was not really familiar with synthetic biology, but I got involved with teaching the undergraduate synthetic biology team that was run at TU Delft, where I started my lab. And, you know, going to the competition with these synthetic biology students, you know, I was just so inspired by just the amazing potential of this field. You know, I saw teams that were using bacteria to make bricks on Mars, to build a moon habitat. There were teams that were, you know, using bacteria to encourage plants to have thicker root growth, to help remediate in desert areas. You know, and just the potential seemed amazing, right? And it got me really thinking, you know, can we use bacteria as a way to replace kind of traditional chemical factories or materials production factories that are responsible for so much waste in the world today, and, you know, environmentally unfriendly production techniques. So, you know, the idea of having bacteria be your factories to make materials, I think, has all these interesting implications. So first of all, bacteria are self-replicating, so your factory makes more of itself. Bacteria are incredibly cheap and easy to grow. And literally all you need is, you know, a glass full of sugar water, and then they'll make more of themselves. I love that bacteria, you know, and all biological systems have evolved a suite of enzymes that can, you know, catalyze really complex chemical reactions in ways that are really sustainable, right? So biological reactions take place typically, you know, close to room temperature, standard pressure, water-based solutions. They don't require or produce toxic chemicals or toxic waste. And from the materials point of view, you know, if you compare having bacteria make a material versus, for example, pouring cement, bacteria can deposit the materials on bumpy, vertical surfaces, on the ceiling, right? You can bring them with you to make materials on site. And they also work really well in microgravity. So it's interesting potential to make materials off the planet Earth as well as on the planet. So the first thing that I was thinking about working with my lab is, can we make some sort of biological solution to improve the cement industry? Right, so cement is incredibly useful, incredibly popular, you know, for good reason as a building material. It's really easy to use. You just mix it up and let it harden. It's easy to make, you know, simple shapes, right? And it's got really high compressive strength, right? So if you have your, let's see, like your block of cement or concrete and you push on it, it doesn't deform very much. But you've got a long list of cons, you know, at the same time, right? I mean, cement and concrete are really heavy for the volume of material that you make. You can't get really fine patterns, right? Really like, you know, centimeter, millimeter scale patterns. That's very difficult to do in cement or concrete. For me, the real kicker is that the cement industry is responsible for 8% of the global CO2 emissions, which is really, in my opinion, a problem for future sustainability of construction and just, you know, society in general. You know, when you mix up cement to make concrete, it's very caustic, so it's very high pH, which is, you know, not good for the health of people that encounter it. You know, and even as a building material, right? I mentioned the high compressive strength, right? But it falls short in other areas. So it's got low tensile strength and it's easy to fracture, right? So probably everyone's familiar with driving along the road and coming across potholes or cracks in the pavement, right? And this is the reason why when you build a building out of concrete, you know, first you pour the concrete and then you add a bunch of kind of metal bars, rebar into it to make it stronger, to kind of combine two materials together to improve the tensile strength of these materials. And so, you know, you have to do something like that if you want to avoid cracking in your cement or in your concrete. So with all this in mind, you know, my lab was thinking about, can we look to nature to find inspiration for making a material that would replace or even improve upon the properties of cement and concrete? So we looked to natural materials such as seashells, nacre, made by animals. So nacre, that's the name for the inner layer of seashells made by many species of mollusks, so oysters and mussels. Nacre is also known as mother of pearl, or, you know, pearl mother in many languages. So as material, nacre is really interesting because it's almost the same thing as cement or concrete. So it's 95% crystallized calcium carbonate, cement. However, if you compare the mechanical properties of nacre with cement, they're really different, right? So, you know, if you compare cement with nacre, the fracture toughness the ability to resist being cracked of nacre is up to 1,000 times better, despite the fact that it's almost the same chemical composition. So because it's incredibly tough, there's a lot of groups that are trying to see if they can make nacre-inspired materials in the laboratory. As far as I know, everyone that's taking this approach is using traditional chemical approaches, which tend to, for example, require expensive machinery. They take a lot of organic solvents. They make a lot of waste that has to be disposed of. So my idea was, can we use biological approaches to make this amazing tough material in a way that's easy, inexpensive, and environmentally friendly? So here's what we have to make. So this is, you take in my lab, we took the shell of a blue mussel that our postdoc ate for dinner one night, cracked it in half, and we looked at the cross section and took a picture of what it looked like with our electron microscope. And what you see is, mostly we see the calcium carbonate. So we see there's these little thin platelets of cement that are in there. And these platelets are glued together, kind of like a brick wall, by organic sticky polymers. And these are kind of similar to silk, but they're really sticky and they stick the platelets together. So this really intricate nanostructure is at the heart of the amazing fracture toughness of nacre. So if you try to, poke on it, then if you measure the stiffness that way, it's very stiff. So it's hard to, change the shape by poking. So that's the stiffness we have of the calcium carbonate. But if you try to bend it, then it's very resistant to bending, very resistant to fracturing with the bending. And that's because, if you think about the different materials, if you bend your nacre, the cement is not going to bend, right? Cement doesn't bend, but the polymers are squishy, right? So the polymers deform, and that dissipates the mechanical energy you're putting into it. So the polymers deform and your whole material avoids cracking, right? Or, if a few of the layers crack, that's okay, because it takes quite a long time for the cracks to propagate. Like one layer cracks, the crack goes over, the crack goes up and over. So the crack has to go a very long way before the material breaks in half. Okay, so if we're going to get a stiff, tough material, we need to replicate, first putting down layers of cement, and then secondly, we have to glue them together with some sort of sticky polymer. All right, so to solve the first problem, can we make cement with bacteria? For this, there's a bacteria that lives just in the wild in puddles outside that has this amazing property that if you mix it together with a simple molecule called urea, it'll break the urea down to make ammonia. Ammonia is basic, so that'll just, by changing the pH, that'll start spontaneous mineralization of calcium carbonate. So this picture here I'm showing you is, we had just a solution of our bacteria. We put a piece of plastic in there. In this case, I think it's a piece of glass, just some surface on which to layer concrete, and added the urea and let it go. And a few hours later, the surface of our piece of glass was coated in a thin layer of calcium carbonate crystals. So we made a thin layer of cement with this bacteria. All right, so now that we can make cement with bacteria, we have to think about gluing it together. So for this, we used different bacteria called Bacillus licheniformis. This is found living on the feathers of birds for some unknown reason. This bacteria is very familiar to many people who live in Japan because they use it to make a fermented soybean dish called natto, which I'm showing a picture of here. So in this picture, the soybeans have been incubating with the bacteria for a few weeks, and all the stretchy, goopy, stringy stuff you see is a certain polymer called polyglutamate that you can see is like a really nice, sticky, stretchy properties, right? So we thought that would be a good polymer to use to stick together our layers of concrete. So in this experiment, we took, a very small culture, like a 20 ml culture of this bacteria, grew it up. When you add, a nickel metal, you add ethanol, then the polymer drops out of solution. You can kind of stir around and pick up this giant goopy like gumball of polymer. So you make tons of this polymer, and it's really easy and simple. So our overall approach to make our material is we take, just a surface, piece of plastic or glass, we put it with the one bacteria to make a small layer of calcium carbonate. We take it out, we put it in the bacterial polymer to add a thin layer of polymer. We go back and forth over and over to build up a layered structure. So here's an example of an experiment where 20 times we lay down a layer of bacterial calcium carbonate, but we didn't add any polymer. When we do that, we can take a material, crack it in half, look at the cross section, and we see we just made like a slab of concrete, right? So we just have a layer of calcium carbonate, kind of like your sidewalk outside, right? In our other experiment you can do, if this time we're alternating laying down the calcium carbonate with the polymer, building it up like a seashell would be built up. If you look at the cross section of this material, it looks really similar to natural seashells, right? So we see really beautiful layered structures, built up, and we get back just about as many layers as we try to put into it, right? So we put down 20 layers. In most places you can count, 17, 18 layers. All right, so it looks just like natural nacre, but what you want to see, it doesn't have the toughness and the stiffness of natural nacre. So we did a number of experiments to look at the mechanical properties. Here's one, kind of visually appealing experiment. So this is called the 3-point bending experiment. So you take your material, you rest it on 2 points to support it, and then the third point of your 3-point bending is you push at it from the top, and you see how well it can avoid breaking as you deform it. And we're comparing here our layered nacre structure, nacre material on the left side with just the kind of slab of concrete we made by bacteria on the right side. And as the experiment goes on, you see the two materials behave very differently, right? So on the right side, when we only just had just the layers of calcium carbonate, we see that really early on in the experiment with low forces, cracks develop and quickly propagate. They spread through the whole material and the material is just cracked in half. Whereas on the left side, our layered nacre material does develop micro cracks, right? So at the end of the experiment, you can look really closely at your material and you see, we see very small cracks that appear, but they don't propagate. They don't spread through the material. So this is perfect. This is behaving just like a natural seashell would behave, in which case that, cracks form very late, they take a lot of force to form and they don't spread rapidly through the material. So overall, if we're looking at the properties of our material, so we're starting with these polymers, the organic matrix that is neither tough nor stiff, and then we're interspersing it, between some bricks of calcium carbonate, so either aragonite or calcite, which are much stiffer but not that tough. And then here I'm showing you the properties in green of our bacterial nacre, which you see it's much improved in toughness compared to either the organic matrix or the calcium carbonate, right? So, and I'm really excited that our material is as tough as many different types of nacre. So, many different types of mollusks will make seashells. So we see all different toughnesses if you look at natural nacre. And ours is as tough as many or most of the different species of seashells that you'll see out there in the wild. We have quite good stiffness. It'd be great to see if we can get even stiffer to approach that of natural seashells. So I'm really excited that our lab has shown that we can use only bacteria to produce 2 different materials. One of them is our composite, our layered material that's really tough. And if it's interesting to someone, we can also make just straight bulk calcium carbonate that's more brittle. So reducing our material in really macroscopic amounts. As far as artificial nacres go, ours is the stiffest ever created, and it's as tough as natural nacre. In addition, it's very extensible, so you can bend it very far without it being brittle. And everything is made, very eco-friendly, water-based solutions. And instead of releasing carbon dioxide like the current cement industry, our process is carbon fixing. So it's actually a net positive for global carbon emissions. We have lots of interesting ideas for what we can do with this material. So since nacre it's not only tough and stiff, but it's tough and stiff for its weight, right? So it has high specific toughness and high specific stiffness, which I think is really interesting, for example, for making components for airplanes or, spaceships or naval ships, right? Because in these situations, you want to be sure that your vehicles, your vessels are as lightweight as possible so you can save on the cost of rocket fuel or, fuel to transport your vehicle around. Natural nacre has an interesting property that it's anti-fouling, which means like stuff doesn't grow on it. You don't get fungus or bacteria or even, you know, barnacles growing on natural nacre, which could be interesting, for example, for kind of outside or in the water applications for building buildings, or for example, statues. We want to look nice outside without a lot of maintenance. I love the fact that our nacre is fully biocompatible, right? So calcium carbonate that we make the platelets out of, that's similar to what's in your bone in your teeth, your bones in your teeth. And then our sticky glue is, you know, edible, right? So everything is fully biocompatible. So I have this idea that could we use this technology, for example, to make implants like bone grafts, for example, if you've, you know, broken or chipped your bones, or even, you know, nowadays, many people that break their bones, you have to have one surgery to insert a pin to stabilize your bones while they heal, and then a second surgery to remove the pin, right? You know, I imagine that, you know, potentially if you use a pin made out of nacre, then that could just be fully incorporated inside your bone and not, you could save yourself the second surgery. You know, and finally, I love the idea of, could we use the technology to make tough structural materials on the moon or on Mars or various outer space colonies, right? So in my opinion, you know, if you're going to go and make a moon habitat, make a Mars colony, I don't think you're going to want to fill your space shuttle full of concrete cinder blocks, right? Because that's going to be super heavy, take a lot of rocket fuel to blast off into space to escape Earth's gravity, right? So instead of putting cinder blocks in your space shuttle, I think you should put a tiny tube of bacteria in your shuttle, so that once you get there, you can, you know, thaw your bacteria, mingle it with, for example, the regolith, like the dust on the moon or on Mars, because that has lots of calcium in it. And I think that you could potentially use that to kind of cementify the moon dust and make a brick out of it that way. So we have lots of stuff we want to do with this project in the future. So we have students working on tuning the mechanical properties. So if we can make our layers thinner, our material should not only be tougher, but also for the first time be iridescent, you know, look pearly like seashells do. We're also working on, can we tune the flexibility of our materials? So seashells, nacre that we're inspired by, you know, it's very similar to what eggshells are made of. That's also calcium carbonate mixed with polymers, except that the interesting thing for me is that eggshells can really vary in how flexible they are. So like a bird egg, like a chicken egg, is really stiff, it's not flexible. But a reptile or a frog egg is also made with calcium carbonate and polymers, but just a different ratio, you know, more polymers, less calcium carbonate. And that's very flexible while still being tough. So we're definitely looking into, can we change the ratio of polymer to calcium carbonate to tune how flexible our material is while keeping our amazing toughness. We're certainly looking to see if we can make thicker samples faster. It'll help for scaling up and speeding up production. We have various collaborations working on to see, you know, how corrosion resistant is our material, is it anti-fouling like the natural nacre is, to test if it would be suitable for applications outside the lab. You know, and of course we want to see, can our material have a shape, like a three-dimensional shape? Because, you know, materials have shapes to, you know, fit the particular use you want them to have. So this is the whole bucket of worms right here, right? So how are we going to make materials with bacteria that have a shape, right? So when people grow bacteria, it's either, you know, shaking around in some liquid solution where everything's mixed together, right? Or it's growing on the surface of kind of like Jello, right? On the surface of just some agar plate, where over time your bacteria spreads out and your shape, whatever shape you started with, just dissolves, right? It like loses resolution. So my lab is working on developing new technologies for patterning bacteria, so depositing them in 3 dimensions. This is the work of these amazing undergraduate students I taught at Delft several years ago now, where we developed the first ever 3D printers that print bacteria. So here's a picture of my team, my students. We won the grand prize at this championship, at this competition back in 2015 for this idea. Our students have developed the, as far as we know, the cheapest bioprinter ever developed. And it's fully DIY, so anyone can make it themselves because our printer was made with children's toys called Connects. So it's a really fun tool that I think could be great for, you know, educational purposes, and you can make your own if you want to ask me or for advice would look at the paper. You know, but I really love this idea of 3D printing a bacteria and using it to deposit bacteria in defined 3-dimensional shapes. So my lab is still working on it. We're developing beyond the toys that we began with to make it a real kind of industry ready technology, right? So here's what our printer looks like. So now that we upgraded from the toy beginning, it's slightly more expensive, but as far as we know, it's the second cheapest bioprinter ever developed. It's about $500, you know, 450 euros to make, and it's much more precise than the toys that we started with. The basic idea is you have a culture of liquid bacteria, and it's in a syringe. The syringe is in a syringe pump, this red device in the corner. Then the syringe pump, you know, squeezes the piston of the syringe, so it squeezes your bacteria out the syringe through some tubing over to the print head. So we took a commercial 3D printer that's, you know, meant to print plastic. We kind of ripped off the extruder that extrudes the plastic and replaced it with our tubing. So now instead of depositing melted plastic, we're depositing our liquid bacteria onto the surface. And so the great thing about this is that, you know, we can control very precisely how fast our syringe is being squeezed. And we also control where the print head is and where it goes to. So we can deposit our bacteria in kind of almost any arbitrary 3-dimensional shape. So this is really, I think, kicking off a totally new revolution in bacterial technology and in making, you know, making products with bacteria. So I see tons of applications for this, right? That I think people can start to develop, you know, all over the globe, I hope. So one idea is, so my lab and others are looking at, can we develop standardized biofilms? So biofilms are when bacteria in the wild, they often don't live as individual cells. They live as a community that has a lot of structure, right? So this community has like more bacteria at the bottom and less at the top. So it's a great way to make kind of like a biofilm on a chip that can reproduce the 3-dimensional structure of biofilms so that we can use this to develop and test new drugs that kill bacteria. So the next generation of antibiotics might be ones that target biofilm bacteria, like the ones that cause cavities in your teeth. There have been some great ideas, for example, for using 3D printed bacteria for purifying water, right? So imagine you can print your bacteria, maybe some shape like a pound sign, you know, something with a lot of surface area. And then if your bacteria are trapped inside this printed structure, then you can move your bacteria. Like if you can put it into one pot of water, have it clean up the contaminants, take your bacteria out, put in a different pot of water. So you move your biomass quickly and easily from place to place to clean up you know, anything contaminating the water or even to extract, right? So bacteria is, for example, responsible for the bulk of mining of copper ions from many copper mines throughout the world. And this could be a way that you could, you know, easily extract your copper from the mine tailings, and then it's isolated in your structure that you remove. You know, my interest is very strongly towards, can we, for example, make environmentally friendly biomaterial like our nacre that now has a shape. So if you print the bacteria in a shape, then have it make nacre in that shape, now you know your nacre has a shape, right? You're not only limited to just making kind of the same type of material throughout your printed structure, you can also make a gradient, right? So you can like lay down more bacteria here and less there. So material is kind of more stiff here and less stiff there, for example. So can your nacre be any shape? Another great idea that other, you know, groups have thought of is, can you have, for example, personalized skin grafts? So the Stuttgart lab in Zurich had this great idea that there's these bacteria that make a lot of cellulose. That's kind of like a nice resilient polymer. And they were able to use technology inspired, I think, by us to, they deposited their bacteria in a 3-dimensional shape, kind of around like a doll's face. And then the printed bacteria made a meshwork of cellulose that, you know, had the same shape as the doll's face. So this could be a great way to, for example, make a skin graft that would really match, you know, the shape of your body part that needs the skin. Could you make, for example, you know, you print materials that respond to environmental cues? So for example, in response to a changing environmental stimulus, could your material become tougher? Could it heal itself? Could you detect, could the bacteria in your material detect contaminants in the environment and then activate circuitry to remove the contaminants. There's this amazing group that develops the idea of living tattoos. So they printed bacteria in kind of a really thin, flexible format that could go on a person's skin to detect certain, sensors, certain signals from your body, and then respond by producing a signal, right? So could this be a way, for example, to monitor your body's insulin or glucose levels for diabetes patients? So these are just, I think, a small list of, I think, the amazing revolution that we can have in depositing bacteria in three-dimensional shapes for, a wide range of applications in society. All right, so just to briefly wrap up, so I talked today about my lab's efforts to use bacteria to make structural materials inspired by seashells. We're also making living materials with our 3D printer to work on engineered biofilms and other applications. We're also using bacteria for many other applications in my lab. We're making conductive graphene-based materials. We're making bioglass for optical materials. And we also have several projects where we've been looking at using bacteria in combination with Mars regolith or moon regolith to make materials in space. So here's my research group, and I'm happy to, start a conversation with everyone about this. Shall I stop sharing my screen?
Yeah, thank you, Anne. This was lovely. Very much appreciate that. You keep that stage for us, and I would say CJ was right, it didn't work, and I would love to go into a Q&A. So probably let's start with Aranveed. Unfortunately not here anymore, but he asked how easy it is to prototype of such a cement from bacteria.
How easy
Could you hear me?
Is the question. So I feel like this reaction of producing the cement with bacteria is one of the easier ones we ever did, and it worked great the first time we tried it. So, we've had success at all different, scales. So from like a small, milliliter level up to almost a liter, and we're able to deposit our cement on almost every surface we tried. We didn't have great success depositing it on metal surfaces, but on lots of different plastics or ceramics or composites or glass, we get a nice layer.
Okay, and also what type of, for example, you use materials for cement, how easy is it to skip them in different...
Malte.
Yes.
For me,
yeah?
you are very hard to hear.
Yeah, same.
For me, you're very hard to hear.
Oh.
So maybe allow me to pick up for a moment if my connection quality is better, and until you find a way to fix yours, and continue with the questions. I will just pick up one of the other questions that I noted, and this one is from Patricia. And Patricia, maybe you want to unmute yourself and ask your question about what other materials might be possible to produce.
Yeah, hi. Yeah, are there any other materials than cement you can grow with that? I can imagine that some polymers might be possible also to grow with bacteria. And maybe, if I may, I would like to expand the question if you can make some kind of combination of materials then.
Mm-hmm. Yeah, I mean, bacteria make tons of materials, right? And some things you can find species that naturally make materials, and other things you can engineer them to make materials, right? So I just mentioned a few things here. We can make this mineral, this calcium carbonate mineral, that we can make these sticky polymers that are found in the Japanese food. There's bacteria that can make cellulose. Bacteria have been engineered to make silk. There's, a lot of other minerals that people are working on. So we're working on making bioglass, so like silicon, like polysilicate materials. Various engineered bacteria can make other sort of metallic oxides, right? So we've shown that we can use bacteria to make magnetite using simulated Mars dust. So there's a lot of stuff that you can make, right? And like my approach is typically, if I have this idea, I want to make something, like it's possibly already happening in nature. And if it's not, and there's a lot of enzymes you can kind of combine and engineer your bacteria to produce that will make something that you're interested in. What was your second question? I'm sorry.
How you can combine it, because I imagine if you have two species which you need to exist in parallel, but which maybe have different cultural conditions or cultivating conditions. I don't know how could you solve that problem, or just alternating or whatever?
No, it's a great question. Yeah, so right now we're just alternating, which is fine as far as it goes. But yeah, like we're looking into ways that we don't have to do that, so we can make more of our material faster, right? So like one thing I think is interesting is the idea of to make it into a two-step process, right? So if you look at how a snail builds their shell, right? They don't do layer by layer like the oysters and mussels do. Snails do like a two-step process, right? So they'll make like a thick layer of polymer first, and then it's the first step, right? And they'll mineralize it as their second step, right? And there's so many different polymers out there, we're trying to see like which ones are compatible with this process to see if we can make kind of a snail shell that's going to be still quite tough and stiff, but is, more able to be made in like a bulk process. Yeah, and just in general, I think that making kind of consortium, like bacteria consortium that live together in harmony as different species is, I think, a really kind of upcoming area in synthetic and applied biology approaches. And there's, papers coming out all the time and people are trying to see if they can get yeast and bacteria to live together or different bacteria species.
Thank you.
Before I hand back over to Malte, Patricia, I would like to also give you a moment to elaborate a little bit more on the practical implementation that you are planning, that you are looking at. So what is the product you're working on?
Yeah, I'm about to found a company with my co-founder about toxin-free and durable ship coatings under the waterline. So we're actually pretty interested in preventing fouling or making it as hard as possible. Let's put it this way. And yeah, it's very interesting.
Yeah, so as soon as it's springtime again, we have a reservation to do some anti-fouling tests at a dock where basically we're gonna make a big piece of nacre, drop it in the water, and then see what grows on it over a few weeks. So yeah, I hope those tests go well.
May I add a short question? What is the surface like when you make samples? I can imagine when you're doing cement samples, it's pretty rough.
I mean, it's like kind of bumpy. Like often our last step is a quick polish, and then, it's pretty easy to polish it and make it smoother. But the bumps are really on kind of the micro scale. So.
Yeah, that might be enough for some microorganisms to settle there when you want to prevent fouling. Did you already do tests? I didn't quite understand.
Oh yeah, so these tests, we have to wait till it's springtime again. We have a reservation in April or May to start these anti-fouling tests.
Yeah, maybe it would be very interesting to hear more about that, at least for me.
Yeah, sure.
Thank you, Patricia. And Harman asked, how do the mollusks create nacre?
Yeah, so when mollusks make nacre, they do it all by themselves, so they don't work together with any other species. But it's the same layer by layer process that we're doing, that we were inspired by, right? So it's actually like a decent amount of calcium in seawater and many different types of, natural water supplies. So they let in a little bit of seawater, and they template the crystal growth. But they'll have this kind of very complicated, so we have a much simpler version, right? But the mollusk version is a very complicated combination of polymers that kind of resemble silk or chitin or cellulose, mixed in with some proteins with negative charges. And so this kind of, having like regularly spaced negative charges templates, where the calcium ions bind to start growing the crystals, right? So like nuclear crystal growth. Yeah, but then they'll make a little gap. They'll make a layer of nucleated crystal growth, and they'll lay down another layer of the proteins and the silk, and they'll just do that over and over again. So it's really a layer by layer growth, like we do in lab.
Okay, great. Thanks for answering. And also, do they work like in a symbiotic relationship with natural environments, mollusks in general, or?
So they don't work together with any other organisms to make the nacre. There is actually interesting evidence. This is a bit for the specialists maybe, but so the different types of calcium carbonate crystals that you can make. And so there's evidence for, like in the lab, we can see that if you have more magnesium present, you get one crystal form over the other, right? And you can see if you look at like when different mollusks evolved, some of them evolved when there was more magnesium or less magnesium in the ocean. And based on that, like that determines like what crystal form they have, right? So there's evidence that like based on kind of the ratio of salts in the ocean, that changes the crystal form that you get. And some crystal forms are tougher than others, which is interesting, right? So one of our other next steps is we're trying to see if we can, change the salts in our reactors in the lab to change and get more of the tougher crystal forms to make a tougher product.
Okay, great. And CJ asked, how much human intervention was needed creating the layered structure?
Yeah, so what we do is like, if this is the thing we're making our nacre on, we'll put it in the one bacteria culture, put it on a layer of concrete, take it out, you rinse it off, then put it with the polymer and then back and forth. So yeah, we have to transfer back and forth for every layer, but then the bacteria does all the work after that.
And does it actually need human interaction, or could you also use like a robot hand or something like that?
Definitely use a robot hand to do it. And that would be great in the future.
And what's the time scale? So asking like how many hours did it take to build like a new layer of bacteria? You also mentioned that, I guess, in the presentation for us, at least.
Yeah, sure. Yeah, so putting a layer down, it was great because you don't have to do anything. You just put your piece of plastic in the bacteria and leave it. So you just usually the experiment we just leave overnight, just because you don't have to worry about it. If we cared to make it faster, which I think we do, you could do that in maybe 2 hours. So we can have 2 hours to build a layer of cement, rinse it off, maybe like 10, 20 minutes to put the polymer on, and then back and forth. So you could put, several layers a day.
And are there actually like not natural boundaries, but like boundaries on scale? So could you use like, I don't know, 100 square feet of like let's say a glass or container or something like that?
Yeah, so we've definitely been working with expanding the surface area of the samples that we're producing. We haven't gotten as big as you're suggesting yet, right? But yeah, we're scaling up. The important thing is going to be, right, you want to be sure that, for your concrete layers to grow in a reasonable amount of time, you're not going to want to have like a really massive sheet of, for example, like a massive window pane sheet of glass. You can't have a single crystal starting to grow in the middle to expand to cover this whole thing, right? The important thing is you want crystals to start growing all over the surface so they expand and bump into each other. So for that, all you have to do is make sure that your polymer is distributed. So in our case, like our sticky polymer, that's what makes all the nucleation sites. So you start the crystal growing all over the surface, right? So if your polymer is evenly distributed, then it should be making crystals starting to grow all over the surface as well. And then I think you should be able to expand like quite easily.
Okay. And I'm a little bit curious because I read in the morning the paper you've written 4 years ago in the Netherlands. And I'm curious about what are the new milestones you had during the last 4 or 5 years within that field? And where do you think could you be in the next 5 years with your research?
Yeah, so we're pushing really hard on the idea of, first of all, can we scale up? So can we use this kind of snail approach to make a thick layer of nacre in a really fast 2-step process? So scale up. Secondly, can you make a shape? I think as soon as we can get this two-step approach working, then the next step is going to be, to 3D print our polymer and then mineralize that. So then we can have nacre in interesting 2- and 3-dimensional shapes. Not currently possible. And then the other approach is going to be sticking with our layer by layer approach to deposit it. We're looking to see, can we make it tougher? So if we make the layers thinner instead of thicker, we'll have, I think, the toughest nacre ever produced. And we also have opportunities to make it look reflective and iridescent, right? Which is interesting for a lot of people, not only for its kind of optical properties, but also for its beauty, which will be great. Because, for example, when this nacre paper came out, I actually got a bunch of emails from people that, for example, they decorate guitars with mother of pearl. They make these traditional drums that are covered in mother of pearl, but seashells only come in certain sizes and shapes, right? So if we can make kind of artificial, beautiful mother of pearl in any size and shape you want, that's going to be really valuable, I think, to kind of artistic people as well.
Very true. Also for industrial purpose. I think Herman has another question. Shall I unmute you? Hi.
So my follow-up question was asked rather by my Maltese just now, in terms of how quickly and how big could you make these, largely with the perspective of replacing concrete in the construction sector. And would we possibly be able to make structures that are 100 meters high, for example, like an average skyscraper in any large, densely populated city? So do you think that's feasible?
So this is an interesting question. So I have two answers for this. So there's a couple of companies that are starting up that have a great approach using the same bacteria that we use to make our concrete, right? So for us, we're looking more at like a coating that's very thin and tough. We're kind of like a thicker coating. For like really bulk scale, people are looking at this technology. There's a company called Biomason in the US where they're literally making bricks, like a big brick, using bacterial mineralization, the same bacteria that we use, right? I'm also collaborating with the lab, Will Schubar's lab at Boulder, Colorado here, where he has an interesting approach. It's a different bacteria, but same kind of chemical reaction where he'll mix it with sand and then it'll basically cementify the sand together, right? That's another way to make a big scale. They just dig the sand out of their riverbank. Their approach is interesting because what they do is they mix together the sand, the bacteria, and then some gelatin, right? And what they showed was that you can, after you make the brick, you can, heat it up. So you melt the gelatin and then the bacteria are still alive in there, right? So you can take your brick, ship it wherever across the world, melt it, grow up more of the bacteria, and then make 10 times as many bricks, right? So it's really like a self-regenerating brick. Okay.
But, linked to this, the question I asked you earlier was about how the mollusks create the nacre. And is there a possibility to, there was talk of a robotic arm, but is there a possibility such that to stimulate these bacteria to create an extruded structure? Is that possible? So that it grows in the shape or form of the end product that you're looking to create?
Yeah, so our approach for that is we have, active experiments right now on this where, so we're 3D printing different hydrogels, something kind of like, if you're familiar, like alginate or agarose or gelatin or something like that, right? Kind of like something that water can penetrate, right? But still holds a shape. And then we're printing bacteria inside this, this shape, right? So we're printing bacteria inside this kind of gel that has a 3-dimensional shape. And then the next step is we have to make sure that we get mineralization throughout this shape, right? So some gels are not chemically compatible with this reaction to make the mineral that we're discovering, but other ones seem like they're working okay, right? So this is, I think, really interesting to see. Yeah, if you kind of print your bacteria inside a scaffold, then have them mineralize the scaffold, I think that's going to be really interesting.
Right. So when do you see all of this coming to market in terms of actually being utilizable?
So just like the plain bricks, that's already on the market here in the US, this company Biomason. the group that has like the living bricks that regenerate, they're just launching a company right now. And the 3D printing, you know, I hope in like 2 or 3 years maybe.
All right. Thank you. Thank you, Anne.
Haman, do you also work in this kind of field? Because I'm curious, where do you see the, if you have seen the presentation right now, where do you see like the biggest space for yourself? So like in terms of industrialization, because you're asking a lot about like cement and those kind of things.
Yes, so I always look at finding a practical and a practicable solution to existing challenges. So my background is in engineering. I don't have a life sciences or a microbiology background, but I did study a bit of material science, and in the larger sustainability argument, the way we use, extract and use materials is frankly unsustainable. I think that is something we'd all agree on. So I've been looking at finding solutions to the basic simple problems that create massive amounts of waste around.
Can you give some example?
So for example, plastic blisters for pills and commonly used medicines, or packaging materials or thermal insulation materials, things that are not very complex or high tech, but are produced in very large quantities and create huge amounts of waste that end up going to landfill and which are frankly non-biodegradable. So it's more in terms of finding these answers, and that's why I was curious about the construction sector, if there was a possibility of creating a material that could be produced in large quantities and could, to an extent, replace cement or substitute it to some capacity. So that's essentially where my focus is. But my day job is in waste management, in fecal sludge management. Mm-hmm. But again, it's the space that we are at is a combination of biology and engineering. So that's where I work in, essentially bringing the engineering perspective to trying to scale up fundamentally what is very exciting new age research all over the world. Mm-hmm.
Okay, great. CJ, you also asked about like the human factor. May I curious what type of work you're doing, and do you see some symbiosis between your work and the work of Anne?
Yeah, ZJ's has some great papers in the field recently.
Oh, hey, thank you. I was really impressed by the EM photos you showed. It's like textbook level. I'm like, that's mind blowing. Yeah, really amazing. So I was just like wondering if one day you can see us like replicating that kind of layer structure, just like by the bacteria themselves. Like that would be really awesome.
This was one of my first grand ideas when I started this project, right? Was could we have some sort of, I'm sure you're familiar with like the repressilator, like the oscillating genetic network, and could we have something like that would, have automatically periods of turning on the urease expression to make the concrete, and then you turn that off, then you turn on the polymer production, right? And I think that, these genetic circuits that make the oscillating behavior, they're getting better and better over time. I don't know if they would really be, independently able to lay down 50 layers, right, without getting out of sync, for example, at this point. Chemically, it's a bit of a challenge, right, because the reaction that makes the crystallized calcium carbonate goes to really high pH, right? So it goes up to pH like 9.5 or 10. That's why you get the mineral forming, right? Mm-hmm. So I think that, even if you were to have sort of built-in genetic oscillator, we'd still want to have something to like flush the solution, I think, because you want to, every time you have to like have this pH change again. With that in mind, there's other enzymes that seem capable of more kind of localized calcium carbonate mineralizations. So we're also, there's a lot of stuff we're, hoping to launch my lab in this project. This is kind of one of the more side projects at the moment, right? But if there's other enzymes that can nucleate calcium carbonate crystals without this crazy pH change, I think that might open the door to really having like a one pot solution with, either, either some sort of like one bacteria that oscillates the genetic circuit, or could we have, for example, like light inducible systems, right? Where you have the bacteria living together, you shine blue light and turn on making the calcium carbonate, then you shine red light and turn on the polymer and alternate that, right?
What
These
would
are all
mean,
like much farther future, I think, ideas.
but I'm not curious. So what would be mean if you say this is a one-pot solution for like, you could use it for different industries as well, or what we, because you were speaking about like, okay, what could be like with graphene, what could be in like space tech, biofilms, whatever. Is a one-pot solution means, okay, we just need one solution and then we can expand it to different industries as well? Or is it always with like adjustments?
I think you get to tailor it for your application, right? I think like this in particular one-pot solution would be for the problem of making a layer by layer deposition approach for the material, you know, and that's gonna be a big challenge to do that all in one pot, but we can certainly work towards that. We're trying different chemistries out. But then, yeah, I think, you know, for every every type of material, for every shape or particular like way that you're tuning the components, you're gonna want to have like a different setup, like a different way that you're turning the bacteria on, for example.
Okay. And CJ, because Anne read your paper, I'm very, what's like what type of industry solution you're working on? And is like Anne's solution like in symbiosis with that and you can do things like together?
Well, yeah, we do have like one part of our work is trying to build a co-culture of two species so they can have this kind of division of labor. Mm-hmm. For example, like we recently created a type of material that's made from kombucha mother. So basically,
are you that paper with Tom Ellis? That's such a cool paper.
Oh yeah. We
worked
together
like yesterday or something.
The day before. Yeah. So the concept is like some bacteria that are really good at producing cellulose, just like Anne mentioned before. So we just let them focus on that part because apparently engineering those bacteria has been like difficult, at least for now. But at the same time, we incorporate in another species, which is the baker's yeast that we know how to engineer pretty well. And there are like tons of genetic tools, and it's actually one of the major workhorses in the industry. So we incorporate that species and put it together with the bacterium. So together they make this synthetic kombucha mother, where you can produce a lot of cellulose by the bacterium. And the yeast, they can be engineered into biosensors. They can produce enzymes. They can, for example, like Anne just mentioned, like you shine blue light, they produce some like bioluminescence enzymes. So they are kind of like a Polaroid film kind of thing. So the goal of this project is actually we actually work with E. coli biofilms before, and the problem with E. coli biofilm is like that's not a lot of materials. A lot of measurements and characterizations was done under a microscope. And we thought if we want to bring the lab system into the real world, it would be nice to work on something that's already producing a lot of materials. So that's why we turned to like kombucha mother. And we found like in that way, you can actually produce quite a lot, maybe not to the factory industry level, but a lot of material that can be like grown just in like on your kitchen table in your glass or jar or mason jar within, I think 3 days would be enough to produce a really thick material. Yeah, so that's what we are trying to push for.
Yeah, kombucha, if you guys aren't familiar, I think it's a really great resource for these kind of biotechnology approaches because if you let it ferment under certain conditions and for a certain period of time, you get a delicious drink, right? But under other conditions, if you just make your kombucha and let it sit on the bench top, it'll grow this thick layer of cellulose on top, right? Which is not something that like the home cooks necessarily ever experience, right? But it makes this like fantastic material because when it grows this layer of cellulose has these really long fibers that are really like intertangled with each other. So it's like a really tough material actually, right? And it's very flexible. It's always been tricky to work with for my lab as well. You guys I think have a great solution because it's super hydrophobic. So the fibers are so entangled that you can't really add something to it once it's produced because it repels water, right? So I think the disease approach is so fantastic because they already mix in the yeast from the beginning. So the yeast is, like, as far as I understand, incorporated in while it's growing. So it's already in there and it can already respond even like once it's produced.
And what are you using it currently for, CJ? Like you doing like for materials, like for example in the textile industry, fashion, or where can you use it usually, like from the kombucha?
Oh, so apparently there are a lot of designers around the world that have been using the kombucha mother as an artificial leather. Mm-hmm. They can make clothes, they can make different kind of like product design using those things. So on top of that, if you add some enzymes or like yeast to produce pigment, you can add color to the material. You can add some functionality, like potentially they can self-clean themselves to a degree. So that's what might be possible if you use that kind of material in fashion, mm-hmm. so to speak. But in other like directions, some battery companies were actually like asking us if you can do some like interesting things with bacterial cellulose, because bacterial cellulose is already used in batteries, some of them.
Oh, it is?
And
for battery storage or for energy storage, or what type of batteries?
I think it's just like a good separator or something. I'm not really familiar with how this, like batteries work, but yeah, it's used in like some of the batteries out there. And it's because the physical properties of bacterial cellulose is naturally better than the cellulose produced by plants, right? Because it's very pure cellulose. They don't have impurity like lignins and other things you might find in plant-produced cellulose. So in other directions, like bacterial cellulose is very biocompatible. So a lot of groups using them to produce tissue engineering scaffolds. So we were thinking like maybe it would be cool if you have the yeast there. They're already making a lot of human growth factors and other like medical related protein or peptides for us. Maybe you can do like a one-part reaction. You can actually incorporate those things into a tissue engineering scaffold. That could be useful for some like medical applications. Yeah, so another thing is like for food, right? Obviously, there are already people using kombucha mother as fake beef jerky kind of thing. I personally haven't tried that yet, but definitely you can add some flavors into the yeast, because there are a lot of things you can do with the yeast. For example, like a very common question we receive from the audience would be like, can you incorporate the yeast that's producing CBD into this thing so you can like spike the kombucha with like, you know, yeah, really functional drink, basically. Yeah.
In our lab, we're interested in these like tough structural materials. So, one of our first ideas was when we had the idea to make, snail shell two-step kind of a process, was we did like what ZJ is describing, and we grew some kombucha. Literally, you brew some green tea, add the kombucha, you grow your thick cellulose layer, and we added that. To that, we added the bacteria that make the minerals, but we couldn't get any minerals inside because, it's so hydrophobic, like nothing penetrates. But we have something hopefully coming out soon, under review, where we take the thick cellulose layer, we literally just throw it in the blender to chop it up. Then we can mix it with the bacteria that make the minerals. And then you get this kind of like thick slurry, kind of like a paste or something, and you can mold that in different shapes, right? And once that mineralizes with the bacteria, we get this like super interesting material. So my student, he makes it into like a bowl shape, and then he can turn it over and stand on it without it cracking, or he dropped it from the top of the building in Delft and it didn't shatter. So I think that's pretty exciting. So hopefully that comes out soon and people can read it.
That's really amazing. Can't wait to see that happen. But is pH a thing for you guys for doing the engineering? Like, is it challenging? Because the kombucha culture is pretty acidic, right?
Yeah, we kind of rinse it off.
Okay.
Yeah, we rinse away the pH, the original pH culture.
And could you actually combine both of the technologies? Like, let's take kombucha for layering and then layer top your bacteria, and then you have like a new, let's say, brick instead of a cement brick.
Yeah, it's an interesting idea, right? I think the important thing, for if you want to have mineralization, right, is you have to have like these nucleation sites that have hopefully a negative charge, right, to line up the positive calcium ions. So you know, cellulose is pretty neutral. Like it doesn't have the nice charges like we'd want to have like a nice nucleation site within our nacre. But we often talked about like, do we want to like functionalize it, like, you know, add negative charge groups onto it, which, you know, at that point, I think we're starting to have a lot of human intervention that goes against our original approach of having something easy and cheap. That's why we went to this other polymer, like from the Japanese food, right? Because that already has the nice charges that we need to nucleate the mineral growth.
Great. Thanks.
Yeah, I would just like to say, sorry, like food is really a great inspiration for material science, especially when you're using biological systems. Like the system Anne just described is just amazing, super sticky. Maybe you can use that for other adhesives as well. And then kombucha and then sourdough is also a very interesting system. Yeah. Hopefully someday we will see that kind of like living materials coming up.
Interesting. Has anyone else some questions? Matthew, Harman again?
I have another question. The first question I forgot last time. You said, if I understood right, that it might be possible to grow the material on site. For example, if you want to make some kind of additional layer on a house, that you can grow them there. How do you make sure that the cultivation conditions are okay? Because I just know from the lab that you have your small plate, put your culture on it or on agar or whatever, and put it in the climate chamber, for example.
Yeah, the great thing is this bacteria is easy to grow, right? It's bacteria that naturally just grows outside in puddles. So when we're in the lab, we don't even put it in necessarily like a really warm incubator. Like it does like quite well at room temperature as well as at just slightly over room temperature. So you don't want to, you know, to be outside when it's, you know, like 4 degrees Celsius maybe, right? But if it's kind of like a nice, you know, 15, 20 degrees Celsius, that's already, I think, plenty warm enough for it to do its job. And then, you know, like the other tricky thing is you want to make sure that when your bacteria is growing, that other bacteria don't also grow and take over the culture, right? So in the lab, you know, we start with like very pure, like sterile culture ingredients, but to a certain degree also, like you don't need to worry too much about that if you start with like a big injection of your bacteria. Then if you know, if it starts with mostly your bacteria of interest, then it's harder for other bacteria to take over, right? So, you know, like once you start your initial like starter culture, then like your production culture doesn't have to be all that sterile actually.
But I think in the end, you will have to meet some quality standards with the material you produce there. So how could you prevent irregularities there? For example, let's say you need some a certain thickness of the coating you make on the wall. How do you make sure that it's everywhere the same, or at least that thickness?
Yeah, so like the first thing I was talking about was if you want to do it, you know, like rough and ready outside or something like that, what are the possibilities? If you're trying to do something like to a certain standard, then I would say you should make it, you know, in your factory, in your lab, right? I mean, the good thing about this is that, you know, it takes place in like a water-based solution. So things will mix really well. And I think as long as your, you know, your polymer or whatever you have that's making the sites for the crystal to begin growing, as long as that's like evenly distributed on the surface, then your crystal growth should also be quite even.
Yeah, okay. And what about the cost?
The cost?
Because I can imagine if you want to compete with cement, for example, which is not the most expensive material, as I know.
Very cheap.
Yeah, I mean, so for example, like, I mean, Biomason, like the company that makes the bacterial bricks, they have quite competitive costs, actually, right? You know, it's very easy and cheap to grow bacteria. Like literally all you need is like sugar and water and you grow your bacteria up very well, right? So, you know, I think, you know, bacteria is something that grows really well at scale because, you know, everything grows in just like a water-based culture, so it mixes really well, and you don't need to worry about necessarily, you know, when you scale up, dead pockets or something like that, right?
That's
So-
great. So the main argument there would be that it's more eco-friendly than the standard production, I suppose.
Mm-hmm. Yeah, I mean, so I would say it's eco-friendly. You know, it's carbon fixing instead of carbon releasing, right? It's something where, you know, for like concrete, you have to worry about, you know, you only grow it, you only deposit it somewhere very flat, right? But your nacre, you can grow like also on the underside or something vertical, right? Microgravity, you should also be able to deposit it.
That sounds promising. Thank you.
So probably my last question for today. What are materials that are already existing out there that could be placed like industrial standards, like cement? Patricia was talking about like airfoiling things. So what are currently like industries that we could replace today by alternative or regenerative materials?
Mm-hmm. Well, there's a lot of, I think, interesting ideas, right? I think a lot of like medical implants could be replaced by something much more biocompatible that's made by, you know, like bioprinting of, you know, human tissue cells, or by, you know, like producing, like ZJ was mentioning, the cellulose materials that are really very biocompatible. Or a nacre could be, you know, a bone implant solution, right? You know, I think CJ had a great list already. So food packaging, I think, is an interesting idea, right? Because you can make something that is, you know, very like tough, but like different degrees of flexibility and very non-toxic. I think it's biodegradable, so better for the environment, like Harman was talking about earlier. Let's see. I think that there's a lot of interesting ideas. For example, could you make you know, beauty products, right? So, you know, nacre is very reflective, you know, because calcium carbonate has a very high index of refraction. So could it be something you like? So for example, many makeup companies will buy artificial pearls made in factories that have like mica cores, right? So not made with the really biocompatible, like shell type of materials that natural pearls are made of, right? So if we could, instead of using these kind of these artificial pearls that have mica and heavy metals, and instead use something more like what our lab makes, right? You could have these reflective materials in your makeup that, you know, conceal your pores without having the tax on the environment. Let's see. I think there's a lot of interesting ideas for various coatings. Like, can you replace like paints or other building materials, you know, with biocompatible or bioproduced objects?
Great.
Yeah, and the whole world,
like,
the whole world of like the vegan foods, right? So like vegan milk, vegan cheese, vegan meat, vegan leather, right? There's a lot of, you know, possibility to replace animal products with stuff that yeast or bacteria produce instead, but that have the same biological composition as the materials that inspire them.
And is it like growing vegan food? Like, for example, CJ also mentioned like kombucha and food and alternative meat. Is that also like that you could just, I'm thinking like, okay, we want to have like alternative sushi or salmon. Is that possible to coat like in a kombucha or whatever material you want to have?
So there's a company in Canada, I forget their name, but they're trying to make, you can say if you call it vegan or not, right? But they're trying to make fish where you don't catch a live fish or farm a live fish. You have kind of fish cells in a dish and you can grow a piece of fish that way. So this is already a product in development in Canada.
Yeah, I know. Yeah, I know them. They're called finless leaps. But okay. So any other questions by the audience? If not, we will. Okay, so for us, the next steps are, first of all, thank you everyone. Thank you, Anne. Thanks for having a great conversation. The next steps will be, we will connect like all the people together that they could like exchange further ideas or connect to each other. And what we would love to see is that actually from NextNess we have like an community on foundation where we can actually start like not working together, but like expanding all those ideas and scale regenerative materials. That's what we want. And we connect like with researchers, investors, startups, and all those kind of people. And we're planning the next event already, and we would love to get like feedback, what we could do better. And also like if you have someone in mind who could be one of our next guests, I would love to speak with them as well.
Cool. Thanks for this invitation. It was a great discussion, and it'd be interesting to keep more in touch with the guests as well as the organizers of this event.
Great. Maybe some last words by Thomas or Stephanie, because thank you also.
No, I want to thank you all. It was a small round, but it was super interesting. Listen to all of you, and I have the feeling that there were some sparks created in between. So I'm really grateful for that, and yeah, we will follow up by providing this recording and connecting and letting you know about our next steps. So this is a journey. We have no clue where we're going, to be honest, but we're having fun along the way.
Exactly. Thank you, Anne, so much for preparing all of this fantastic work, I think. And I think we all agree that we wish you all the best for the future, and we hope to have you back.
Oh, great. Thanks so much.
All right, everyone.
Thank you.
Malte,
Okay.
you're going to close the round?
I'm trying to figure out how to close Zoom here, but yeah.
Have a wonderful day, everyone.
Thank you,
Yes.
everyone.
Take care.
Ciao.
Have a good
Take
day.
care. Bye.
Goodbye.